Okay, we'll get started. So the goal for this week is to teach you about how to do mapping, how to, once you get data from DNA sequences, how do you figure out where does it align and what is its sequence composition. But before we do that, we have to finish what we left in the last class. So if you recall, we had talked about second generation of sequencing. The idea there behind being that you have either a set of molecules which you're going to flow through a slow flow cell, which has a very high precision camera. It's going to be able to look at what bases get incorporated at each step, and that's your essentially read through. Today we'll just very quickly talk about what's broadly called the third generation of sequencing. The primary aim of the third generation sequencing technique were twofold. One is to make it faster. The reason second generation was slower is because you have a lot of wash cycle where you saw molecules coming in, getting attached, the polymerase waiting, and then another round of cycling that has to happen. We will see today if that can be done away with, right? So each of this extension was happening by synthesis. This is called sequencing by synthesis, or SBS method. That's your second gen of sequencing. So before we talk about third generation sequencing, we'll just do a quick overview of what we heard in the last class. So broadly, Illumina, which is the commercial name of the second generation sequencing broadly, which has survived, we start from a genome, we perform a DNA fragmentation step. These set of steps are broadly called the library preparation. Your library consists of adapters ligated to a shared DNA fragment sequences, right? So this is your DNA library that gets prepared. Now somebody will take this library, will throw this off on the sequencer. This step is happening outside the sequencer. The ligation of the molecules is happening outside the sequencer. Once you throw this off on a sequencer, you'll have probes attached to it, which are complementary base pairs to these adapters. And because of each spot having exactly the same adapter and being able to do biochemistry reactions, these are very small fragments when you talk about this scale. They will make a lot of copies here, which is called a bridge amplification. And this will end up doing an artificial inflation of these sequences, making such clonal copies. These are called clonal because they originated from the same clone, right? And this is what your sequencer is now going to read through. What does the sequencer look at? So once it has these copies, your bridge amplification is complete. You will now add all the you'll add the DDNTPs, which emit a fluorophore. At each point, you're adding all four bases based on what gets incorporated. So in this case, T is getting incorporated. In this case, G is getting incorporated. In this case, C is getting incorporated. This is one cycle where I've added one base. I will do a wash. This washing will get rid of the fluorophore. At this point, I have a camera which has recorded what is being emitted. And once I've done that, I'm going to repeat this cycle again after washing. So when I wash, the fluorophore gets washed out. It can get extended. I add another set of these molecules, and this extension keeps happening. So if you look at this, if you have a camera, this will appear as, if you have ever seen fireflies in the night, this will keep emitting signal which is equivalent to a firefly emitting a light signal at each spot. Okay. Any questions about this part? Okay, so broadly, that's what is second generation sequencing, or Illumina sequencing to be very precise. When you get the data, we will talk about read 1 and read 2, and once we do the hands-on Thursday, it will become clear. But there are two ways to sequence. So one is called read single end. I'll just call it SE. In single end, the read that you get there is only one read that comes out for each sequence. That's called just read 1. In paired end, you get two sequences. One is read 1 and the other one is called read 2. Read 1 is not a complement of read 2. It's not a reverse complement of read 1. Don't let anyone just tell you that. There are people, unfortunately, who think like this, but that's not correct. What it is you have a DNA fragment that you're sequencing. So if you recall, when we got this on our flow cell, we have a polymerase sitting which is going to do the extension. So if this is your 5 prime, this is your 3 prime, this is the 5 prime, and this is the 3 prime of the extension that is happening, right? So in one round, you only usually get one of these reads. Okay, so I have a choice that I can make this copy and I will be done with. I can wash off this particular fragment and then I'm done. But in a second case, what I can also do is I have another round where once I make this copy, I can get rid of this. I can ask them to make one more round of clonal copy such that I get a bridge like this. Once I do the wash off reaction, it's going to wash off the first base that formed the bridge, and I keep the other copy of the bridge. In other words, once you make a copy and then you let the extension happen, you can retain the copied version of it. And at this point, your 5 prime and 3 prime have exchanged, right? Because when it got bended over here, so if I make a copy of this sequence here, this will bend over here and I will make a copy in the other direction. Now I'm looking at this copy where I have my 5 prime, I have my, if I just do some abuse of notation. So if this is 5 prime, 3 prime, for this to make copy, now I have my 3 prime here. So this is my 3 prime and this is my 5 prime of the bent over sequence. Okay. So now I have a choice that my polymerase is now going to sit here, because again, for the extension to happen, it will always happen in the 5 prime to 3 prime direction. I can now sequence from this end. So while I got my read one, because my polymerase was sitting here by hybridizing, I have another choice of sequencing, which is to sequence from the other end of the fragment, right? So if this is your fragment, I can either sequence from here, or once I make a copy of this, I can sequence from the other end, right? This is what gives you a paired end of the read. The hope is if you have enough length of the sequence, they will merge somewhere in between. They will start to overlap. So if your DNA fragment, for example, is short, read 1 and read 2 can overlap. But in other cases, or in most cases, you will have segments which would look like this. This broad thing that you're trying to sequence is called, the fragment that you originally sheared is called the insert size. This is the size of the sequence that you have inserted in the flow cell, and this is your goal. But read lengths are often fixed, so you will only get 100 nucleotides or maybe 150 nucleotides or maybe 300 nucleotides from one end and from the other end. This will remain largely unsequenced. But the reason it's not a problem, why is it not a problem? If you're not sequencing the middle part of the fragment, is this a problem or is this not a problem? I'm saying that we will never be able to sequence this. Okay, you can map it, fair enough. So you'll map it, you'll get mapping here and you'll get mapping here. What about the portion in between? But what if my biological changes are in that part? What if I have a mutation here? Okay, so very good. So when you did this part of DNA sequencing, if you recall, there is actually one hidden piece of information which I didn't talk about, is when you're making these cuts, you're making a version of it where you have multiple such copies and you're going to make these cuts randomly. Right? So even when I started from this one fragment, we're not actually starting from one fragment. You have a bit more copies to do away with. And when I add my cuts, I will have all sorts of cuts possible. So it's not that one DNA sequence is there is only one copy. There are actually multiple copies that you're shearing it on, you're cutting it on. So you get multiple such cuts, and that's why, some point it will happen that you will also have reads. Right now the read length was this. I will have reads which will look just like I will have fragments which would just look like this, which in other words will try to capture the entire part of your genome. Right? So ideally, you'll make enough copies, you'll make the shearing such that, and there are like some numbers out there which allow you to do that, but that's what essentially you do to control for this artifact. Okay. So everything is happening in a single strand version, right? Like wherever, yeah, you're forming the second strand, right? You just wash off the single strand. You just wash off the single strand, right? Like, and that part, how exactly it is controlled for that, I'm not super clear on. But when you are doing this wash off reaction, you're just washing off the original copy of this read one. So if you go back to that video, at some point it actually features up. So now when you try to connect the dots, go back to that original video and see it. I don't have it here, but if you search, if you go to the original site, you will see that video, and now it will make more sense as to what exactly is happening. Okay. Any questions? Okay. So remember this, that while these read one and read two are not complementary pairs, they are actually sequencing the same fragment of the DNA but from different ends. And why should I care about this? Like, why should I do this? So the read one and read two will always come like this. So if you have one FASTQ file, we will talk about what FASTQ files are. They will always come paired in the sense that if there is an R1, there will be a corresponding R2. So they go hand in hand because they represent the same molecule. And there's an identifier for each molecule that goes, that is thrown off to a sequencer. But why should I do a format which looks like this versus the single end version, which is just also called FASTQ1, where there's only R1? What kind of errors? So even if you get incorrect base faults, it's fine, because in that scenario, honestly, you're increasing the chances of your errors, right? So if the error rate for this is E1, this also will be E1. You've actually increased the error rate. Yeah, okay. So how does this help actually tell you where you're getting there, but not fully there? How does it tell you where it came from? Where it came from? That's the important question. Where we came from, where it came from, that is what we have to figure out. There's something which I'm not telling you, told you about the genome yet. And that's why it's not an obvious answer, but you're there, almost there. So why is this problem going to be slightly easier than solving this problem? It's a needle in the haystack problem, which means that one read, where did it came from this 3 billion base pairs, which part of that genome did it come from? I have no clue. Okay. But why would it help if I have an anchor? Which is the second read? Why should I do this versus? I'm telling you, I'm just forcing you to think that paired end is often better than doing single end. But it's also comes at a cost, because you're going to have more money thrown in here because you're sequencing something extra. But why should people? Okay. Okay, so very good. So if there are repetitive sequences, which I've actually not told you at all about, right? But if you've taken BB101 or any other similar thing, you would have probably come across sequences which could look like, this. They're just, random base pairs or random motifs which are repeated all over again, copy pasted. But if I have a read which is, let's say, so this is one such sequence, but it occurs in multiple locations of the genome. Now, if my read is TATA, if I just do single end, I will not be able to tell you where did it come from here or did it come from here. These are two different locations in the genome. But if I do a paired end version of this, I will have another anchor which will allow me to go and sequence. While I only sequence this particular part, maybe the other end will read this. And if I know this, I can confidently say that it came from here. Well, in this case, I did a mistake, but let me make this G. Right? It came from here and not from here, right? Because ATAC only occurs here, and the fragment between them is common, right? This could have come from anywhere, but once I have my anchor, I can confidently tell that it only maps uniquely here, and cannot map to this location. But if I had only one read, I will have to discard it because I cannot confidently tell that it come from here or here. But this anchor, which is a second read pair, if I have it, I can confidently tell that this was the first shear and this was the second shear, right? Or this was the entire fragment. This is read 1, this is read 2. Okay, is that clear? Okay, so that's called paired end or mate sequencing. Single end is very cheap, very fast. You can do it actually very overnight. Modern versions. Paired end, it doubles your read cycle because you have to read the second molecule once more. But it's very useful if you have a repeat region to resolve it, right? And if what is a repeat is not clear, you can think of this one portion being repeated in your genome multiple times at a different location. There are like bunch of these things which make mapping problem much harder, which we're going to talk about today. There is some technicalities, this you should be aware of, which you will come across, maybe you will come across in hands-on, but you will definitely come across when you're doing your projects, which is, if right now we have been talking about samples which is coming from one individual. But what I could do is I could take the sequences of all the 24 of you sitting here, I could pool their DNA. Before I pool this DNA and I want to do a whole own sequencing for the entire class, I will add another set of adapters which are called your I7 and I5. As I had told you, like how did these name come about to be is not very clear, but this is some version of barcoding that they were inventing. These are called barcodes, just like you have barcodes in your grocery or any other item that you buy nowadays. This barcode uniquely identifies the sample. So for example, if I take Bipra's sample, if I take, remind me your name again, Rajat. If I take Rajat's sample and I want to sequence both of them together, but I still want to be able to tell them that this came from Bipra and this came from Rajat, I can confidently do that by first taking Rajat's sample and then taking Bipra's sample, attaching a specific green and orange, which is specific to only Bipra, a green and orange, which is specific to Rajat, putting this and then pooling them together and putting this on the sequencer. Because once I do this, the next set of, when I start to read their sequences, I will read the sequencing, the barcode as well. So this will become an integral part of your read 1 and read 2. But it will happen only at certain location because it will happen only, you will know in this case, the first, for each shear of the fragment, let's say this is B and this is R. For each shear, if I have already given them some set of identity first, right? So let me make this with a different color. So now these are identical, otherwise identical sequences, but now they have their own identity. If I now put them and if I start sequencing now, I will be able to confidently tell because I know the sequence belongs to B and this belongs to R, and I will be able to read it off. I will be able to confidently tell which came from where. Okay, this concept is broadly called multiplexing, which allows you to pool different samples together and put them on the sequencer at once. So you don't have to wait if you don't have, for example, enough DNA. So the reason it's often used, for example, there are projects like the Human Genome Project, which we'll talk about, but also in the Indian context setting, recently concluded project was what's called the 10,000 genomes. You can read about it. It's called the Genome India project. where the idea was we'll sequence 10,000 individuals from India. If I want to sequence 10,000 people, I will not have access to 10,000 sequencers, and I don't even need it. I want to be able to do it in such a way so that I retain the identity of all those 10,000 individuals, but at the same time get their sequencing, because sequencers are limited. Maybe you're talking about like 5 or 10 or maybe 50 sequencers at max. If I want to run them at the highest capacity, I should be able to pool all the data and put them on sequencer overnight, maybe run it for 3-4 months or maybe up to a year, and then at the end of the day you have 10,000 sequences, right? So usually in a country which is just beginning to sequence, you will take at least 1 year, it will just take to warm up. So if you even get 5,000 sequences in 1 year, that's a win. And this is how you do it, by multiplexing, so that you can use the maximum capacity of your sequencer. Okay. Okay. The text is just for your reference later to read, but that's the broad idea for you to understand. Okay. These are specific DNA sequences. So you can do those calculations. These are calculations have been already done that they don't they don't have overlap with your sequences in the genome. Right? So you can induce either one kind of repeats, or you can make it very specific so that it only matches one location. The same way you would do when you're designing primers for a particular guide or a particular gene, or primers for a particular target sequence. You can design them. And the easiest way to do this is just to make them longer, so that chances of colliding with any other sequences is very, very hard. Okay. Okay. So one problem, yeah. No. So once you pool them, you have already done that ligation reaction. So you've already put the sequences. So it doesn't matter whether even DNA fragments from 2 different people, they can come to the same flow cell as well.
But then light, like 2 different lights at the same
time. No. So when you make that at the spot, that's a unique DNA fragment, but it doesn't matter where it's coming from. So when you get this at the spot, right? When I put this at each spot, everything is a copy, because you start only from one strand at a time. This, whether it came from B or R, does not matter right now, but this will be unique. And the copies that you get out of this, let's say this is from B, it will always be copies of B. But the next spot next to it can have copies from R. You could use lesser number of the flow cells, but often what will happen is your flow cell size is fixed. I could have, let's say, I could take, for example, 10 picogram or 100 picograms of DNA sample. That's its capacity, right? But from one person, you're only able to get like 1 picogram. So you would ideally are underutilizing the capacity of that flow cell, because the amount of reagents are not going to change based on how much amount of DNA you load. It's still going to load the same amount of reagents. So you'll end up wasting these reagents, and as you'll realize, these reagents are the ones which are costly. So that's why you don't want that to happen. You would rather wait to maximize putting the amount of stuff. It's a good question. I should have motivated it that way. That you're trying to save money, because the amount of resource you're going to spend on the reagents is going to be the same, whether you load 1 person's DNA or 10 person's DNA, as long as it does not meet the upper threshold of the sequencer. You would want to wait till you get enough capacity of it to load, so that you utilize all of them together. Okay. Okay. The problem with this particular regime of technologies is there's a rate limiting step, because there is a delay which is caused by the need to remove the 3' blocking fluorophore group after each nucleotide addition. This is the wash cycle that we talked about. So because there is wait cycle, this will be slightly slower. It's not extremely slow. You could finish, for example, a modern, like I could send my DNA sample and in one of these versions, maybe on this one, or actually on this one. This is the highest end of sequencers. I could probably finish my 1 pass of what's called 5x genome. We will learn what that 5x means in just a bit. It's called the coverage. Maybe overnight, maybe 24 hours to 30 hours, right? You can in principle finish this. Okay. Ishan, I'll have to ask you to leave if I see both of you. Last warning. If I don't say something, doesn't mean that I cannot see. I choose to ignore, but there's a limit to everything. Okay, so there's a third generation sequencing. The idea behind third generation sequencing is to overcome some of the challenges of the second gen sequencing. The idea there being that right now we are limited by the length. The length was, as you saw, 150 to 300 nucleotide, and the time, the time to throughput, you cannot do this in real time, for example. Can we do the better? Can we increase this length? Longer is better in terms of sequencing, because if you have a long sequence, the chance of this repeat getting confused will be lesser. If I had originally sequenced the entire length of fragment here, there will be no chance because of making an error in terms of mapping, because it will only map to one location uniquely. The reason I got this problem was because I could only sequence 4 bases, because this was second gen. So can we do better? So the first version of technologies that made it possible was called the PacBio. This is again the commercial name of the technology, but the principal concept on which it is based is called real-time single molecule sequencing using PacBio. It gives you reads which are 1 kilobyte to 20 kilobytes, right? 1 KB to 20 KB. Now, in fact, even longer version, which is possible. And it gives you in real time. So I can actually, as the molecule goes out, the sequencing can be read on any modern computer. The idea behind this is very simple. The way to avoid this addition is to get away with this blocking group, right? If you somehow can get rid of this idea that why are we blocking? Because I want to give the camera enough time to process what just happened. But if I could have a very high precision camera again, which could read in real time that what's being incorporated, I don't have this problem anymore. And that's what this particular technology tries to do. For this, what you have to enable first is you have to circularize the template. So rather than having a long DNA, you have circularized it, and it will go through what's called a very small nanowell, right, or a picowell. This gives you enough precision. So if you want anything to be extremely precise, the first thing that you should do is you bring in focus. The focus in this comes because of what's called the ZMW, the zero mode waveguide, where you're working with light as a wave, right? So you're at such a small location that you can actually have a extremely precise control. Each of such well can have a polymerase sitting there. One such example is here. And as the molecule gets incorporated, you're again incorporating the labeled version. So these are again labeled. You have a polymerase setting here. You have circularized the things. This is going to move, by its definition, going to move one base at a time. Based on what molecule it incorporates, it's going to emit a signal. Now you have an extremely precise camera which is sitting somewhere here or at the top, which can look at, because you have enabled this in a very, very small scale, you can measure how much amount of light is being emitted as it gets incorporated. And as you measure that particular light, you can guess, or you can actually predict what is the current or what is the wavelight which is being emitted, which you can control and tell that whether it came from A, G, C, or T, right? Because this is based on the lambda calculations of all of these fluorophores. So for example, green will have a different lambda as compared to T, as compared to C, and as compared to G. You can measure this in real time. Okay. This allows you to do longer because in this case, there is no waiting period for the polymerase. In the second generation, this was waiting because you had one round of these come up, wait, wash, wait, wash, wait, wash. Here, they're flowing in real time. It's going to keep extending, and the measurement is also going to happen in real time, right? Clear? So usually that's how the precision, this is at the pico level, so your picometer level. So usually you will be able to control that internally, which is your only one strand is going at one point, right? So in this case, each of your different wells will have a different version of the fragment which are being sequenced. Okay, so you're optically observing these polymerase. This is optical observation, polymerase mediated synthesis. Last we saw was SBS, sequencing by synthesis. There is a polymerase which was there earlier also. The only difference here being that you are making it much more robust by not allowing it to wait and let the molecules flow in real time without any washing involved. The reason you require this zero mode waveguide or these small wells is to be able to measure light at extreme precision, which otherwise would not be possible if you're just sitting on a glass flow well and having a camera mounted on the top, right? Because there you had to amplify the signal. The same thing, the reason you're able to get rid of is there we made a lot of copies so that we could see which signal is coming. Here I'm saying that, okay, let me go and make this molecule sit in a very, very small room so that the moment the molecule incorporates anything, I should be able to measure what got incorporated, right? So here we had expanded the set of molecules. Here I'm shrinking such that I only have one molecule in a single molecule in my very short room. Here I had a fixed size room. I was expanding the molecules to fill that room so that I can detect it. Here I have shrunken the room and I've installed a very precise camera which can measure in real time what's happening. Broad distinction is clear. So this is the concept. This is the commercial name, PacBio sequencing. At some point 2 years ago, PacBio, let me recall who bought whom. Illumina bought PacBio, but because of some other clauses, some monopoly clause and everything, that was overruled, and now they still exist as two separate companies, but they offered complementary services. In the sense that if you want precision, you would go with Illumina. So Illumina will talk about the error rate. The error rate is very, very small because you can measure, you have amplified enough so that you're detecting the right signal, and the fidelity of the polymerase which is going out of sync is very, very low. In this case, the error rate is actually higher because while we are promising every molecule one camera, that camera itself is not precise. The entire process itself is not very precise. So your error rates can go from 12 to 13%. They have actually developed something called Hi-Fi, which we'll not talk about, which is called the high fidelity version of PacBio, which rather than we were working only right now with single molecule in one well, you can actually make multiple copies of it and work with multiple copies to get rid of that error problem. But we'll not talk about it. That error is now down to like half of this, if not lower. In Illumina, the error rate is 0.1%. In this case, this is 11 to 12%, right? The order of magnitude higher, 2 orders of magnitude higher error. Okay, questions? Yeah. Yeah, so the advantage of this being done in a circular way is that for each molecule, you have enough length of the molecule that you can cover, right? So if I made this linearized, in beyond a point, I can also use this information to tell at what point am I starting to recirculate the same molecule. That information will help me come up with, reduce the overall error load here, right? Because if I start from a linear version, I will be done here. Then I have to load another molecule. On the other hand, because I cannot control the amount of shear that I've done to my original fragment, this will allow you to go to longer lengths. Because even if my entire thing finishes, I will do another round of it rather than having to wait. So it allows you to gives you more power to be able to do those things at scale. It will drop down, right? So if you want to, so that's where the question comes out based on that. But remember that each point you're still spending reagents on being able to sequence the same molecule. So that's a choice that you have to make, that do I want to go wider or do I want to go deeper in some sense by focusing on only few molecules, right? Because you're still wasting reagents, right? They're not free. In principle, you could, right? But some of it is beyond the limit of if it's just being emitted by one molecule, and if there's a systemic error that is happening at each well, that systemic error you will not be able to get rid of, right? If every time that A is being measured with a precision of 90%, like even if you sequence it again, it will still remain 90%. That is not going to go up. You're not getting an independent measurement of the same process, right? So that's not useful to do it. But 1 or 2 rounds might be useful, but beyond that, it won't really help to have like, okay, 90% of the time, So there's a joke. Let me tell you the joke, right? Which is a lot of us spend like our life, half of our life here, right? Which is like 30 years in this service. Now you'll see again. You'll see for yourself there are spectrum of people who teach here, who are standing here and teaching. So there's a difference between an experience of 30 years and one experience repeated 30 times. Okay? There's a difference between an experience of 30 years and an experience of 1 year repeated 30 times. So if I take this course and I teach it exactly the same for all 30 years, I can do that. But is that something going to be more useful versus 30 different iterations of the same course, right? So that's the power that replication will bring you. Okay. Okay, so that's one version. The other one is more famous. It's called, well, both of them are equally famous, but this is something which changed the game in, sorry, there's formatting issue here, but it changed the game to some extent, which is called nanopore sequencing. The idea behind a nanopore was invented, I think, around 1989. George Church. People know George Church? Have heard of George Church? People have heard of mammoths being brought to life. Okay, so there's one person connecting all those companies that will come out. I think this idea came from Colossal Biosciences, if I'm getting the name of the company right. But he runs a lab at Harvard Medical School or Harvard University, one of those two. I think it's HMS. Where he's been a proponent of synthetic biology in multiple sense and has had number of, every person who trains with him will have one of those companies come up. So we'll not talk about him in general, but the original ideas of nanopore came very, very early on when the sequencing was come over to be. It took, however, 20 years to mature. The idea is very simple. If you can come up with a protein, which is this nanopore channel, and make the DNA, which is negatively charged, to flow through it, as it flows through this nanopore, it will emit a particular current signal. If I can decode this signal, I can read what the sequences is in real time. Right? That is exactly what the idea is. You take a nanopore, which is like a channel protein, for example, you're forcing your DNA to flow through it. There is positive charge, there's negative charge on the other side. There's negative charge, there's positive charge on the other side. It will go through because this itself is negatively charged. And as it goes through, you will have current being measured, because based on what is inside, you will have a different charge that gets measured here, right? So this is your DNA. This is your nanopore. This is negative. This is positive. DNA itself is negatively charged. As I flow through it, there will be a flow of current, because different amount of negative charge based on which base is inside will flow through my nanopore. If I can measure this, right? So again, you will just have to get this particular protein, let this sit, and bring in your DNA and just let it flow through it, right? It's very simple because everything is already charged, so it will find its way through to go through it. This is done again. The commercial version of this is called Oxford Nanopore. It's by a company called Oxford Biosciences. The idea here is this current I can measure back to predict what base it is, right? And this was one of those examples where deep learning or your CNNs actually are used in real life in biosciences. To be able to go from current to a single base pair resolution is actually not very straightforward, because sometimes you could have 6 bases, sometimes you could have 8 bases. So how do you tell to the base level resolution that even if I have 8 bases here, that corresponding 8 base pair corresponds to this 8 bases or something else? That is a prediction problem. So from the current here, to be able to tell at the base pair resolution what base I'm incorporating, that's done by a CNN or a convolutional neural network. This is one of the classical examples in biology and broadly in computational biology, where it would not have been possible otherwise, where you require deep learning or convolutional neural networks to be able to solve this non-trivial problem. So that is incorporated in the training traces, right? So I could start from all human genome, like I can share it, where I actually know the sequence. So I can run a parallel, either a PCR, or I can do a full-on Illumina run. I can load the DNA where I actually already know the information. And I can do this 10,000 times and hope that every molecule that's going, I know the sequence already. And then measure the current, right? And this is very simple. For example, you could do it for polymers. You could do it for poly A's, poly T's, poly C's, and poly G's first. Those are the simplest sequences to synthesize. Or you synthesize a pair of DNA, flow this through it, you know the sequence, and you try to predict what it is. So there is some error rate here. This error rate is also higher, right? The error rate in this case is primarily because what you said, which is the algorithm, is not exact. There's some noise, or the efficiency or the accuracy that you get from this neural network is still not 100%. And because of this, this also has a similar error rate as PacBio, which is 14%. So in other words, you're training your CNN, but it's only able to perform till 86% accuracy. Okay, so there are gaps to be filled here. This is an active problem. I think they're still working on solving this. So in this case, you, as the molecule flows through, you need a force for DNA to flow through, right? You want it to flow through it, because as you flow through it, this nanopore, which is allowing your which is a very small set of channel protein, this reaction will accrue a charge difference. And that charge will be emitted in this portion of the nanopore. It's just a very small channel protein, which otherwise you will not be able to get hold of, right? If you just create a membrane which you positively charge and you negatively charge, everything will flow without any order. This gives you the order in which things are going to flow, right? So it will only allow one molecule to flow at a time. No, the membrane will allow you only to have one molecule at a time. Mm-hmm. Because always you'll have, if there is positive, you'll have a negative earth control, right? Which is, so even if, this is positive, you'll have a negative charge somewhere. That is how you balance. Yeah, that's right. So that's, you're getting this from an ion channel actually. These are ion channels, I should clarify that. So you're increasing the concentration of your ions on one side and you're keeping it lower on the other side. Right? So keeping that membrane is what is allowing you to keep that separation explicitly. It's not like you're actually putting one red wire and one black wire on the other side. Right? So that is okay. So there'll be a separation maintained by this membrane overall. Okay. Any other questions? Is it the charge, you mean that charge difference that you have to create here, is that fixed? So I think that is something which will probably be dependent on the overall size of the pore itself, that how much amount of, what is the size of this pore. But let's say if that is constant, you will maintain such amount so that this information, the flow of 6 base pairs, for example, which is usually what it will capture, can happen at once without any hindrance. At least that much amount should be able to go in at particular time, but I don't know the exact calculation on which the positive versus the negative charge is based. What this allows you, however, to do is go long, very long, right? So if you have, on the other side, if you have enough positive charge, your DNA on the other side will remain stable and you can go longer. I can keep going this till 1 kb, 10 kb, and even up to 20 kb, for example. You can keep going longer because there is nothing technically preventing you from doing this. You will have fall off for other reasons, maybe the DNA is not stable, but in this case, there is no technical limitation. The only technical limitation is, whatever you're capturing here in this pore, can you decode it? But it's not limited by any other technical factors, because it could be like 5 kb long to 10 kb long. You don't have to even have to share it. I can just keep going because technically there's no reagents involved, right? There's no reagents. So as long as I'm able to maintain this charge difference, I should keep going. And at some point, you will require to replenish this the moment this goes on this side, because this will have its own negative charge, right? So you'll have to, some way to have that replenishment. As long as you can do that, you can keep going forever. Inside the nanopore, yes. 4 to 6, yeah. That's right, exactly. Yes, so you're creating every time that you're this channel that you're getting, right? Like this is a artifact of what is there inside, and that is emitted by 4 to 6. So which 6? Like, is it ACTG Is it ACTAA? That is a machine learning problem, right? From one pattern, what are the possibilities? How do you reduce the number of possibilities to do what's called base calling? How do I reduce a signal which is being measured at 6 nucleotides at a time to one single nucleotide? That's a base pair resolution. That's a machine learning problem. Better will be this thing, right? So if you can like do technological solutions where your channel proteins are or your motor proteins are even much more smaller, which can only take one base pair at a time, this would be like the best technology that you ever need. It will take lengthier, right? Because you are going to have that for each thing that has to flow, it can only move one base pair at a time, right? You will not be able to read one particular signal at once. Okay. Okay, so that concludes our generation 3. This was used actually. This is one of those. The field in general draws a lot of flack because often people think that it has not really delivered anything which is meaningful, but it did deliver. Like there is, for example, this is a clear case example where you could go to a clinic, you could go to Ebola places where there have been Ebola outbreaks to tell and to test people, to test sites that is there Ebola actually there. Otherwise, you would have to take that sample, ship it to somewhere where there is sequencing facility available. But I can take my laptop and I can take that what this is called MinION. It actually is the size of my hand. This is actually the size of my hand. So I can take the sequencer on the ground and sequence things. And I would know which pathogen is infecting. Right? And why do I want to do that? Because I can tell tomorrow that there's already a Delta variant on the ground, be ready, which seems to be much more severe. Rather than having the patient to come to the clinic, figuring out what is their sequencing, another 2 days wasted. By this time, the outbreak has already happened. So this is something which allows us to do actual real-time public health based on genome sequencing. Okay, so this was used extensively during SARS-CoV-2 and also during Ebola and Zika virus outbreaks in Africa in particular. I think a lot of labs in India also now have it. It's also one of the cheaper versions of sequencer to have. A lot of people in a lot of labs have it, and you don't require actually a lot of facility to do this because I can carry this in my bag. All you require is some reagents, some pipetting and all that, but nothing very fancy, which otherwise you'll not get. Okay, there's one final thing. Maybe I'll skip the Ultima sequencing for today and let me because this is slightly technical and I'll not be able to finish in 5 minutes. But what I'm going to talk about today is very quickly the format of the sequencing data. And when you come back, I'll talk about Ultima. The reason to talk about Ultima was to be able to talk about the $100 genome. So let me show you that, and we will keep the technical discussion for tomorrow. So what is the goal here? Why are we studying this, and why this class about DNA mapping? So if I have a DNA sequence, right? If I could sequence my entire billion base pairs, I will not have any computational biology would not exist because all of this is solved. An ideal technology will allow you to sequence billion base pairs, but even Oxford nanopore or nanopore sequencing is not there. It will give you 1 MB, maybe 8 MBs, but not beyond that. So if I'm getting 1 MB or 8 MB sequences or mega base pairs, how do I tell where it came from? Did it come from here? Did it come from here? Did it come from where exactly? That is a computational problem. And that is what are going to be our focus. So if these are like short segments in your DNA puzzle, I want to be able to put them together so that I can create the entire sequence. So if this is my entire sequence, what a read will get out is these sequences. Now from these sequences, how do you reconstruct the entire thing? If you have ever played a jigsaw puzzle, which I'm assuming all of you would have, this is an idea about how do you put the entire picture if you have very small pieces together. It's exactly similar because some things will overlap. So you know that all of them should belong to the same position. And some of these are like going to be very, very different. So you know they came from very different locations. If you know this, how do you solve this problem computationally is what our focus will be. But before I do that, why are we talking about it, and why is it even possible? So this Human Genome Project, the idea behind the Human Genome Project was conceived in 1980s, where the idea was that if I could somehow know all the billion base pairs that make up the human DNA, I will be able to solve all problems on this earth. And 2026 is like a revisiting, where all the tech bros feel that once you have all the LLMs go wild on all the DNA sequences, in the next 5 years, all diseases will be solved. Right? So only time will tell who's going to be able to solve this. The first thing that we need is a definition of a disease, right? That itself is not known. So whatever they mean by diseases, hopefully we'll see in 5 years whether company X or company Y or company Z solves it. But to take you back to 2000s, so this idea came up in 1980s, right? And if you recall, it was only Sanger sequencing which was present at that time, because you could sequence short base pairs of sequences, 800 base pairs in 24 hours with a lot of error rate. People started to develop, as this project was construed or was thought about, people started to think about what technological innovation do we require to solve this. And all these technologies that you're seeing up to second generation were actually, just like in the Second World War, we saw a lot of technological innovation come up. Similarly, during the Human Genome Project, just because we wanted to solve that one problem, we were not really fighting a war, but we were trying to solve one big problem. We started to think about what technological innovations do we need to do to be able to solve this. That's where companies like Illumina came about to be, or Iron Torrent, or Roche, however it's pronounced. The first human genome was promised to be delivered in 2003, and it was delivered slightly earlier than that. It cost $100 million to finish the entire human genome at that time. And that cost is now claimed to be only $100, which is what one of the companies claiming that is Ultima sequencing, which we'll talk about in the next class. How all of this became possible is a matter of discussion, but a lot of it became possible because, a lot of these, when they were initially thought about, a lot of it was new. So anything which is new will often be costly. But the reason it has become extremely cheap now is because we are able to do multiplexing, and we have reduced the amount of reagents required for a lot of these steps, which otherwise was not possible earlier. So this was delivered 3 years ahead of time because of FOMO. There were two companies, there were two people who were competing for it, or two groups rather. What you see, the gentleman on the middle, of course, requires no introduction. The gentleman on the left, we lost him this year, is the founder of a company called Celera Genomics, Craig Venter, and the person on the right is Francis Collins. He was the director for NIH till I think 3 years ago or 2 years ago now. The idea behind this project, these two people competing, was everybody had figured out that we have to deliver a human genome where we have to deliver what those 3 billion base pairs look like. But in order to be able to deliver it, we require technology and we require computational algorithms. It would not be possible without computational algorithms. And that's where this third person come in, which is Jim Kent. He was a graduate student at UCSC. He wrote the code for what's called assembling the genome, right? So both of these groups broadly were working with, so his company was working with his DNA, where they had pooled samples from his DNA and like maybe other people as well. And the other one, which was a public effort, they were working with around 30 to 40 people of Caucasian descent, which is broadly Europeans and Americans, where they collected data from blood for females and sperm from males for sequencing. They've again pooled it. At this point, we don't care about which came from what. We are trying to create a genome which would be reflective of all these 30 individuals. They performed this, what's called shotgun sequencing, which is what we studied, which is you chop off the DNA and you study very small fractions of it. Once you get this, you have the computational problem, which is how do you put them together so that they all make one base, right? So if in a sense, this is your DNA, and let's say this is one half and maybe this is the other half, or one part of it. When you sequence something, you'll get fragments which would look like this, you'll get fragments which will look like this, and you'll get fragments which will look like this. So if I told you that this is your DNA, if you knew it, it's very easy to put it. If you knew that somebody gave you the jigsaw puzzle, if I give you that, the final picture should look like this, it's not very difficult to put it. But if there is no picture, how do you put it together? It's a non-trivial problem. And that's what Jim Ken's algorithm tries to solve. It's called assembly problem. The unfortunate thing, however, is we are not going to talk about the assembly problem because it's an extremely hard problem. And I only do easy things. So I'm not going to talk about the assembly problem. We will talk about what's called the DNA mapping problem. The idea behind DNA mapping, this is my last slide for today, is if there are no sequences, which these are unconnected segments of DNA, if I want to put together a full genome, I need to first find out how do they overlap with each other. If these are your DNA fragments, and if you have multiple versions of them, I can start to put them together such that at some point they will start to show some overlaps. Right? So if you had enough of them, you will start to see that this comes first, it shares certain sequence with this half, this shares some sequence with this half, and so on. And by this, if I do versions of this, I can maybe put this together. But we have put this together, which is what Human Genome Project did. So the delivery of this Human Genome Project was this one contiguous sequence, which will become your reference sequence, which is because 99.9% of all individuals, human beings, will have a sequence which is similar to this. This is called the assembled genome. You have assembled it from short fragments of DNA. We will not focus so much on what's called the assembly problem, where you break these DNA and then you try to reconstruct the entire sequence. What we're going to work, however, today is, once you know this reference sequence, how do you tell, when I sequence a new portion of the DNA, what came from where? It's a much simpler problem to solve as compared to putting the assembly problem. So on the left half is the assembly problem, which is generating the reference genome sequence. On the right is the mapping problem, which is given a reference, how do you map it? Okay, I'll end it here and we'll start and resume tomorrow and finish some part of how do you how do you solve the mapping problem. Okay, some of you have not submitted your assignments. Please do, because I'm going to release another one. How was the first assignment? Okay, it is lengthy, okay, but easy, right? It's not very difficult.
Few of them were non-intuitive.
Okay, which ones were non-intuitive?